Mitosis. Mitosis is cell division that is asexual reproduction and is largely used as growth and repair in most multicellular organisms, but is used as the form of reproduction in some others. Before looking at mitosis itself, we need to have a look at chromosome structure. Chromosomes are obviously made of DNA, and that DNA is coiled round small proteins which are called histones. Those are then compressed to form the chromosome, which is this. Now, the chromosome is made up of two chromatids. The chromatids are completely identical. So that means that one can act as a template for another, which will become important later on. And those are held together by the centromere. So the chromosome is made up from sister, that's what they're called, sister chromatids, and the chromatids are completely genetically identical. Now, moving into the cell cycle is made up of a number of phases. We start that, as it's a cycle, there's no sort of starting point, but we have what we call the growth phase, and its technical term is interphase. And interphase, as you can see, takes up the vast majority of the cell cycle. Indeed, some cells will spend almost their entire sort of existence in interphase, and they will only be in the kind of the mitotic part, the dividing part, for a very short period of time. The interphase itself is made up of three stages: G1, which is a growth phase; S phase, which is a kind of a synthesis phase where new organelles are created, and then a second growth phase, and then we move into the mitotic cycle. Looking at interphase in a little bit more detail, there are some key points that go on in interphase. So we have our nucleus, we have our nucleolus, we have our centrioles, which will be as a pair outside the nucleus, and the DNA is uncondensed and just appears as a big splurge. Towards the end of interphase, the DNA will start to condense and chromosomes will begin to become a little bit more visible. So towards the end of interphase, the DNA will condense and you'll start to be able to see the chromosomes. The next stage of the cell cycle is prophase, and prophase is the first stage of the mitotic cycle. In prophase, the chromosomes really start to condense and the nuclear membrane will break down and disappear. So you will start to see the chromosomes sort of appearing as though they are X's in the cell area, and the centrioles will start to migrate towards the poles of the cell. The next phase is metaphase, and in metaphase the centrioles have moved to the poles of the cell and have created the protein spindle fibres between them. The chromosome, which I'm demonstrating here using pipe cleaners, then attach themselves to the spindle fibres and move towards the equatorial plate like that. They're attached by the centromere. The next phase is anaphase, and in anaphase the centromere splits and the spindles are pulled towards the poles like this by the half centromere, and they move towards the pole of the cell like that. Because the chromatids are identical, what you are doing is you're providing a template for each cell. The next stage is telophase, and in telophase what you have are the chromatids at each end of at each pole. The nuclear membrane starts to reform, and the centrioles will replicate, okay, and you will start to look like a cell with two nuclei at that point. Which leads us to the final division, which is cytokinesis. In animal cells, this is fairly straightforward. You have the cell like this, and what happens is you have a furrowing. So you get an invagination of the membrane, and a furrowing occurs through actin spindles, which causes the cells then to split into two. In plant cells, it's slightly more complicated because of the cell wall, but equally it's quite cool. what happens. So you have your cell wall here and you have your cell membrane here, and what happens is that little vesicles containing bits of cell wall migrate to the equatorial plate, and then they secrete their contents into the middle part here to form a cell wall, and then the little vesicles themselves, the membranes of those join up to complete the cell membrane, and so in doing that you end up with two completely separate cells, and that completes the cell cycle.